Tēnā koutou, tēnā koutou, tēnā koutou katoa. Good evening, and ladies and gentlemen and distinguished guests. It's my pleasure to welcome you here tonight. My name's Helen Nicholson, and I'm the Deputy Vice-Chancellor External Engagement at the University of Otago. And I'd like to thank you for coming, and I'd especially like to thank the Minister Woodhouse, for letting us come here and hosting us tonight in Parliament buildings. The 2016 series is presented in association with the Health Research Council of New Zealand as it celebrates its 25th anniversary. And I'd like to take this opportunity to welcome our guests this evening from the HRC. And I know Conway Powell's here, and also Jerome Dowes and Susan Snively, who've supported us during this series. The University of Otago has had a long association with the Health Research Council, and many significant research projects have benefited from funding received from the HRC. And this has enabled Otago's health researchers to take even greater steps in their research endeavours. And I'd just like to share with you a couple of the things, or a few of the things, or most recent examples where we've benefited from HRC funding. So one relates to Christchurch. So Otago's Christchurch Research Heart Institute has been at the forefront of global research into heart disease diagnostics for about 30 years. And it's recently developed a test to quickly and easily detect unstable angina. And it does this by measuring levels of the hormone BNP signal peptide in a patient's blood. And this test is being commercialised through a newly formed company called Upstream Medical Technologies. Meanwhile, in Wellington, He Kāinga Oranga Housing and Health Research Programme researchers are running 6 trials to lift housing standards to improve children's health. This includes insulating and warming 600 newborns' homes in Hutt Valley and Wainuamata, and providing wraparound housing and welfare services to 800 children hospitalised in the wider Wellington region. And then back in Dunedin, more recently, research from the Dunedin's multidisciplinary health and longitudinal study has found that it's possible to detect differences in people's ageing rates, even in young adults. And this leads to the possibility of offering prevention opportunities before chronic diseases become established. Now, there's one person, one member of our Otago academic community who I haven't mentioned yet, and that's Professor Greg Cook. And I'd like to invite the Honourable Michael Woodhouse, one of our Dunedin MPs and a University of Otago alumnus, to introduce tonight's lecturer.
Thank you, Helen. Kia ora, everybody. Welcome back to Parliament, to those familiar faces whom I've seen, and to those of you who are here for the first time. Welcome to what is, I think, Lecture 5 of the 2016 University of Otago lecture series, a series that I have been extremely privileged and proud to host, firstly, as Helen says, as a Dunedin-based Member of Parliament, as a member of the Cabinet that is responsible for appropriating much of the funding through the HRC and directly to the university, and through other means that it gives substance to many of the fascinating bodies of research that are going on, including this one. But of course, as Helen said, also as a proud alumnus of the University of Otago. I guess I have something of a historical interest in this. As some of you know, I was in the health sector for a long time, although not in a clinical setting. The issue of antimicrobial resistance and pathogen management more generally is a very, very live one. It has global interest. The issue of specifically MRSA and resistant strains of bacteria. The most recent example of good primary management and prevention strategies in Havelock North, for example, where we are having to cope with a very nasty outbreak of what's most likely to be Campylobacter through the water system, sheets home, I think, how important it is that we continue to research ways in which to keep our population safe. Before I introduce Professor Cook, can I again apologise? We are in the middle of a very busy parliamentary session, and I'm progressing a rather controversial bill through the House as we speak. It's the dinner break, but I do need to go and do some horse trading with the other parties. Therefore, while I'll be listening to most of the speech, it will be necessary for me to depart before the end, so I'll leave it to Helen to wind up, and I'm afraid tonight I won't be able to join you for refreshments, which those of you who know will be served in the East Wing, and those of you who don't, just follow the ones who do. So tonight's fifth lecture is to be presented by Professor Greg Cook. Professor Cook is a microbiologist at Otago's Microbiology and Immunology Department, another of our fantastic departments. Since starting at the university in 1998, he has mentored and trained over 50 postgraduate students who are our new generation of scientists at the dawning of their careers. In 2014, Professor Cook was awarded the University of Otago's highest research honour, the Distinguished Research Medal, for his lab's pioneering work, which led to breakthroughs in developing more effective drug treatments for tuberculosis. Currently, Professor Cook's work is focused on bacterial physiology and antimicrobial resistance, with the goal of this research being the production of a new class of antibiotics, and we may well need them very soon, termed metabiotics, that specifically target the metabolism of microorganisms. His lecture is entitled "The Race Against Antimicrobial Resistance, Can We Win?" And he'll discuss for us the battle against antimicrobial resistance, which threatens to return us to the pre-antibiotic era, and that is something of a nightmare scenario, I would suggest, as well as how his research is developing new weapons to fight its global spread. Ladies and gentlemen, please welcome Professor Greg Cook.
I just want to thank you, Michael, for that kind introduction, and I also want to thank the University of Otago for giving me the opportunity and come here and talk tonight. So this is the question that I've posed to you. Can we win this race against antibiotic resistance? So if you ask people what was the greatest discovery of the 20th century, most people will tell you it was the discovery of antibiotics. And of course, we all know antibiotics were discovered by Sir Alexander Fleming. And it's been estimated by the World Health Organization that antibiotics on average add 20 years to each of our lives. So very, very important discovery. Antibiotics were so effective when they were first introduced to treat bacterial pathogens that the US Surgeon General, and actually this was the year I was born, 1966, he actually believed it was possible that we'd be able to close the book on infectious disease. That's how effective antibiotics were. So then you start to see headings like this: bad bugs, no drugs. And so you start to wonder, well, actually what went wrong in 50 years? Well, what went wrong is that we treated antibiotics and used them for many, many things. And we actually, the amount of antibiotics that we've used in our hospitals and agriculture is a staggering amount. And in actual fact, various surveys have shown in the last 10 years there's been a 36% increase globally in the use of antimicrobials. In a country like India, we've seen a 62% increase, equivalent to 13 billion units of antibiotics being released. So these are not only being used in agriculture, they're also being used in hospitals. So what happened was, as soon as we introduced penicillin, of course, we had antibiotic resistance to that antibiotic. And so we used to think about bacteria becoming resistant to single antibiotics, but now we've got bacteria evolving to multi-drug resistance, and so MRSA is an example of that. We now have something called XDR, right? So these are strains of bacteria that are extensively drug resistant. So an organism that I work on called tuberculosis, this organism has become resistant to 10 different antimicrobials. And for tuberculosis, we now have totally drug-resistant strains. Now, a point that I really want to make here is that not all bacteria become resistant to antibiotics. So for many bacteria, and an example is Streptococcus pyogenes that causes strep throat, this bacterium has never become resistant to penicillin, and we have no idea why. Right? So we're not talking about every bacterium becoming resistant to antibiotics, we're talking about particular bacteria. Right? So that's the first point that I really want to make. So we underestimated this ability of bacteria to become resistant. Antibiotic resistance is very expensive. About $30 billion a year is spent on trying to control outbreaks caused by antibiotic-resistant bacteria. In the US, 23,000 deaths per year and greater than 2 million illnesses. So antibiotic resistance costs us a lot of money. And if we look at the numbers going forward, this number starts to go up massively. We've been complacent, right? And I'll tell you why we've been complacent on developing new drugs. And so when you see headlines like this, you start to see other headlines like this, that people are now saying that antibiotic resistance is a threat that, could be equivalent to global warming, right? So we need to be thinking about that in terms of which organisms are we really talking about here. Clearly we're not talking about all bacterial pathogens, but obviously we must be talking about some. So the bacterium I work on is this bacterium called Mycobacterium tuberculosis. Right, I'm sorry how this came off here. So Robert Koch discovered this organism about 125 years ago, but in actual fact, Mycobacterium tuberculosis has been around for about 30,000 years. Right? And this organism has existed in a metastable equilibrium with us for 30,000 years. So we never eradicated TB, and TB has never eradicated us. Right? But this equilibrium is clearly changing, and things that change this equilibrium are things like HIV. Right? So this appeared on the cover of Time magazine, right? And it's called Contagion. And what this article was about this bacterium here. So in 2006, we had a strain of TB evolve called extensively drug-resistant TB. So this organism evolved at the epicenter of the HIV/AIDS epidemic, right? So TB, right? So HIV, of the many people that have HIV on this planet, TB kills 30% of them. So HIV individuals are very susceptible to tuberculosis, right? And really HIV was the trigger point for the resurgence of TB, right? We thought we'd wiped TB out in the 1960s, 1970s, and then came HIV. And so what that meant was more drugs were needed to control TB in these immunocompromised individuals. And so it was probably no surprise that extensively drug-resistant TB evolved at this epicenter. This organism was deadly. 52 of the 53 infected individuals were dead in 16 days. If you're HIV positive and you get extensively drug-resistant TB, your life expectancy is about 2 weeks. Right? For drug-resistant TB, we need to treat you for 24 months. So before we can even diagnose it, right, you're dead. This is a very, very dangerous organism, right? And it's now spread to greater than 100 countries. Now you're thinking here tonight, oh, come on, he's talking about countries like Africa, China, Asia. That's never going to happen in New Zealand. Well, in actual fact, we had our first extensively drug-resistant isolate of TB in Dunedin Hospital, 2010. You're probably thinking, well, how does that happen? We don't really have a big problem with TB, right? We have about 1 case of TB a day, and certainly drug-resistant TB. We have drug-resistant TB, but it's not a huge problem. So the key point that I want to make to you is that drug-resistant TB is a shared health problem. Right? We can't sit in New Zealand and ignore what's going on globally. And the reason is that this person that had this extensively drug-resistant isolate was a student studying at the University of Otago. He arrived in New Zealand with a latent infection, so obviously it wasn't detected, even though it had chest x-rays, and he reactivated. It took 66 days to diagnose this student with XDR-TB. So that's the big problem for New Zealand. We're a low-incidence country. We don't have technology to be able to diagnose XDR-TB. We just simply haven't seen enough of this disease. The strain was resistant to about 8 different antibiotics. It took 9 drugs to treat him. He had to take those drugs for 18 months. Cost the taxpayer $180,000. Right? The good thing here is that he lived. Right? If we deported him back to Myanmar, he would have died. Right? And he's still living in New Zealand. Right? So HRC recognised this, that we need to be part of this global fight. So they introduced this thing here called the eAsia Joint Research Program. So my lab was one of the first labs in New Zealand to get one of these grants. And what we did is we partnered with Myanmar. So in Myanmar, right, they have 650 cases of TB a day. Right? They're a high incidence country. Drug resistance is spiraling out of control. Right? So we started this program with them. And so one really cool thing about that is I get to go on road trips with my Vice-Chancellor and my DVC International. So Helen and Harley and I, we went to Myanmar and we actually met with the new president of Myanmar, the lady. So we met the highest person in the country to talk about the research efforts that we're going to do in this country. A postdoctoral fellow in my lab by the name of Tin Ong runs this program. So it's not about white guys in New Zealand telling people in Myanmar how to do research. We want to train the people in Myanmar to do this research, right? They have to take control of the problem, but we want to help them. Right? So that's a great program. It's up and running and it's looking very successful. And what we want to be able to do is to try and get better diagnostics for this organism so we can identify who's got the disease. So antibiotic resistance is obviously a problem, otherwise I wouldn't be here talking. So a guy by the name of Jim O'Neill, as an economist, has released these recent reports. And what he basically predicts, if we don't do anything about antimicrobial resistance, this is how many people are going to die in the year 2050 of AMR. 10 million people, right? What I can tell you for TB, 1.5 million people die of tuberculosis a year. Of that 1.5 million people, 200,000 are killed by drug-resistant strains. What is this going to cost? Well, it's going to have a massive cost in terms of GDP, right? So very expensive, a lot of people are going to die. And so what Jim O'Neill is saying is that we should be moving to a system where that when you go and see a doctor, you do not get an antibiotic until we diagnose you with a bacterial infection. Right? So that's what he's saying. He's basically saying we've got to stop handing out antibiotics like sweets. I think he's taking this a bit too far, unless we can prove that the infection is bacterial. So, for example, TB, it took us 66 days to diagnose this guy with XDR TB. What do? Do we wait 66 days before we give him drugs? I mean, these are the issues that we face going forward. Right? So this is a bit of a problem. So everyone will point the finger at antibiotic use in animals, right? And I've got to be honest with you, 10 years ago, I was one of those people. I was in this very room 10 years ago trying to convince the poultry industry, Minister of Agriculture and Fisheries, to stop using antibiotics in chicken production, right? And Sue Kedgley was my partner in crime, and it's safe to say that we got absolutely nowhere, right? So I actually, 10 years ago, I gave up on this area of research. Right? I couldn't get any funding and it just couldn't get any buy-in. So I walked away from it, and that's when I started working on TB. Right? So this is what happens, right? If we look at antibiotic use in the US, 70% of all antibiotic use is in animals. 30% is consumed by humans. Right? So of all the antibiotics we're using, 70% is flowing into animal production. So let's think about this for a second. Animals get sick just like we do, so they also demand the therapeutic use of antimicrobials. What I'm talking about is subtherapeutic, sub-MIC prophylactic use, right? Growth promotion. That's where there are issues, right? It's not so simple. So here's the important thing, right? So of the antibiotics that we use in animals, 31 are deemed medically important, right? So we've got these 10 here. These 10 antibiotics are currently not deemed medically important. The problem that we've got, because we've got so much resistance, these antibiotics right here are going to become important to us going forward. And one of those antibiotics on there is bacitracin, which is an antibiotic that we work on. Right? Bacitracin is not really used in humans. It's used as topical agents, but it's not used extensively. But people are now saying we're going to have to start using this antibiotic because we're running out of antibiotics for some organisms. Right? So this is the scenario in the US. Right, so this was Sue Kedgley and I getting in trouble, right, because obviously we went after the poultry production because of the antibiotics being used there. I think there was about 86,000 kgs of antibiotics a year used in animals, right, and then so you see sort of stuff like this, superbugs found in chicken dangerous, and we're massive consumers of chicken, 85 to 100 million per year, right, so in order to have this density of farming, you need antimicrobials. Right? Because if one of these chickens gets sick, the whole flock get wiped out in days. Right? And these antibiotics are used for particular things in particular conditions. Interestingly, in some countries in Europe, they've completely banned the use of antimicrobials in animal production. Right? So this is what I came. I came to Parliament, I stood right here, poultry industry sitting right there, holding hands with math. Everybody was in it together, and I basically showed them this data. Right? So we collaborated with the poultry industry. We looked at 147 farms across New Zealand. Right? So we sampled 90% of poultry production in New Zealand. The poultry industry were absolutely fantastic. They were totally engaged in this project, right? So they sent us fecal samples from the animals from the farm. So those fecal samples would arrive at my lab all week, and then we'd take those fecal samples, and then we'd look at the microbes in those fecal samples and test their resistance to antibiotics. And we tested the resistance to the antibiotics that they were using in poultry production. So the antibiotics they were using, right? So the big one was bacitracin, 26,000 kgs a year. Right, these chickens, they always see bacitracin. Some of the other antibiotics are rotated. And so what this table shows, so this was the table I actually showed them. Right, so these are different suppliers, right? So you can guess who these suppliers are. So these are the major suppliers of poultry meat in New Zealand. Right, and what you can see, here's an example of supplier A. So what we did is we looked at 185 isolates. So remember, this is 147 poultry farms. And then of those 184 isolates, what you can see is 183 are completely resistant to bacitracin. Right, so we've got 99% resistance to bacitracin. So I actually said to them, why are you bothering to feed this antibiotic? Because all the bacteria in there are resistant. Erythromycin, used to control community-acquired pneumonia. Right, very high levels of resistance. Right, so this was the one I was trying to get them to stop using. Because this one here, what we found was that the genes for erythromycin resistance were on the same plasmid as genes encoding for vancomycin, which was actually an analog called avaparsin. So we used avaparsin, we fed this to chickens. If you're resistant to avaparsin, you're resistant to vancomycin. And some people will recognize that vancomycin is one of the last antibiotics that we can use to treat MRSA. Yet in these poultry, we had VRE, right? So we had vancomycin-resistant enterococci in poultry in New Zealand. Right? So what we wanted them to do was to stop using this one because it was co-selecting for this. So even when they stopped using avaparsin, the legacy of using this antibiotic, we saw that the VRE persisted. And what this means is that once you get a problem with resistance, even if you take the drug away, it takes a very, very long time for that resistance to come down. It doesn't just come down overnight. And so this was a great example. Even though they'd stopped using avaparsin, the legacy of its use remained in these poultry flocks. And we have no idea where these organisms were coming from. And this was about the time that I basically ditched this area and moved on. Right, so that was the issue. So what is the New Zealand situation? Right, so you saw 70% of all antimicrobials in the US go into animal production. So where does New Zealand sit in this? Right, what's our antibiotic use look like? Well, in actual fact, New Zealand is one of the lowest users of antibiotics in animals in the world. Right, so that's something that we should be very proud of. We're number 4. If we stopped using bacitracin, we'd be number 1. We would be the lowest user of antimicrobials in the world in animal production. Right, maybe that's something we need to. So this is important, right? Ratio of human to animal use. So you see New Zealand, it's a very good number. Look at Norway though. So Norway banned all antimicrobials in broiler production, 37. Right, so just about all of their antibiotics are being used in humans, right? So you can see New Zealand is very, very good, but we could be number 1. You can see some countries, massive amounts of antibiotics are being used in animal production. Right, so that's a very, and then the New Zealand Veterinary Association have come out and said by 2030, which is a really aspirational goal, they want to stop using antibiotics altogether, right? Very interesting goal. I mean, hopefully they can achieve it. The reality, of course, is, so we think about antibiotic use in animals creates antibiotic-resistant organisms, but what impact does that have on human antibiotic resistance? Well, I can tell you, we don't really know. Right? What happens is we have these reservoirs of antibiotic-resistant organisms, whether we're using them in animals or whether we're using them in humans. So this is New Zealand, so this is some work out of Dunedin, and what they basically show is that New Zealand, we are excessive users of antimicrobials. We have one of the highest rates of prescribing in the world. Right? We use a lot of antibiotics in our hospitals. Right? So these are the numbers. Right? So this is doses, daily doses per 1,000 population. You can see New Zealand is right up there in terms of antibiotic use. You can see if you compare us with countries like Netherlands, Norway, much lower. Right? We're not as bad as Greece and countries like this, but we have very high use of antimicrobials. Other surveys I've seen say that 63% of all antibiotic use in hospitals is deemed inappropriate. Right? If you look at this, we're not slowing down. Right? We're continuing to use more and more antibiotics. right? And so, of course, this creates antibiotic-resistant organisms, just like it does in any environment where you use antibiotics. This is some work of Debbie Williamson, right? So this is looking at 2 topicals that you could buy over the counter, right? So here's one called Bactroban. So everybody's probably got a tube of this in their medicine cabinet at home. I know I've got a couple of tubes. Right? Everyone's got one of this. So the problem with that, of course, is that this gets used, it's excessive use. And so what happens is lots and lots of it gets used, and then sure enough, you get resistance. Right? In 2000, greater than 20% of the isolates are resistant to it, so you might as well throw it away. Right? And you can see here in about 2000, they switched to this other one called Foban. Right? So this is fusidic acid. And what you see is we just start using lots and lots and lots and lots of it. Right? And once again, 2013, greater than 45% resistance, might as well throw it away. Right? Once you get these high levels of resistance, then really the treatment's ineffective. Right? So we need to try and stop using antibiotics excessively for things they weren't really meant for, and we obviously need to develop new antibiotics. Right? So the key point here is that we will never beat antibiotic resistance. Right? What we've got to try and do is develop drugs quicker than the organism can become resistant to it. Right? But the reason why we won't beat antibiotic resistance if we don't do something is that, in actual fact, antibiotics have been around for hundreds of millions of years in the environment. Right? Here's a fantastic study that was published in Science. So what this group here did is they just went out into the environment, right, pristine environments that had never seen antibiotics, sampled some bacteria from the soil, brought them back to the lab, and what you see is that, look, we've got bacteria in this soil that have never seen antibiotics that are resistant to 7 or 8 different antibiotics. And the reason for that is that basically just about all of our antibiotics come from soil organisms. Right? They come from bacteria that live in the soils that make antimicrobials to kill other bacteria. Right? That's how we discover antibiotics. Right? So this is not surprising. Other papers are coming out now showing that bacteria can actually grow on antibiotics. Right? So of course it's a nutrient. It's got carbon, nitrogen, hydrogen. Bacteria can grow on it. Right? So we should never be surprised about finding resistance, because obviously the organisms that make these antibiotics obviously already have the resistance determinants in their genomes, otherwise they'd make an antibiotic that would kill them. Right? So all the resistance determinants, all the antibiotics are already out there in the environment. So we need to think about synthetic routes. Right? So it's not surprising that there's all these resistant organisms. Antibiotics have been around for millions of years. Vancomycin, 240 million years old. We've only worked out in the last 50 years that we can actually use it to treat bacterial infections. Vancomycin's a fascinating one. It took 30 years before we saw the first resistant isolate to this antibiotic. 30 years, right before we saw resistance. Other antibiotics, you can see resistance in a year, in a week. Right? This antibiotic was very unusual. The bacteria had to assemble a whole lot of genes in order to become resistant to this antibiotic. Bacteria grow very fast, so if we have one variant in the population, right, so one cell in that population is resistant to the antibiotic, doubling time in 13 minutes, and very quickly the whole population is resistant. Right? Bacteria have lots of gene transfer. There's massive selection pressure, mutation frequency. Right? There's all of these factors that make it very hard to combat resistance. Right? So everyone will say, okay, we've got to stop using less antibiotics. We're going to not use as many antibiotics in agriculture. We're not going to use as many antibiotics in medicine. Right? That's what we should be doing. We should just say we're not going to use as much antibiotics. Well, the reality is, in order to feed the planet, right, over the next 30 years, we're actually going to consume 70% more antibiotics. So we're not going to consume less, we're actually going to consume more. Right? In countries like China, right, they're going to be massive consumers of antibiotics going forward. Right? This is their consumption in 2010. Right? This is what it's going to look like in 2030. As they gear up for animal production, we're going to consume more antibiotics. So what we've got to be thinking, okay, we can try and use our antibiotics better, right? We're going to use 70% more, so what we need to do is we need to discover new antimicrobials. And these antimicrobials, I think, need to be purpose-built. We need to start thinking about using antibiotics in different applications, as opposed to using these broad-spectrum agents that basically wipe out everything. Right? So we need to discover and develop new antimicrobials. Right, so this is what antimicrobial discovery looked like. This was the golden age of Fleming and co-workers. So this is where all our antibiotics were discovered. Right, and so this was the point where we thought infectious disease is game over, so we basically stopped discovering. right? So we had this huge innovation gap with this hiatus from drug discovery. Suddenly we woke up in about the year 2000 and said, oh my goodness, we're running out of antibiotics. And so that's when the medchem people came in and started making derivatives of various antibiotics. Right, but you can see we had this massive innovation gap. Right, so we got complacent. Right, but not only did we get complacent, and even when we started to discover new analogues, it's very, very difficult to discover a new antibiotic. Right? First of all, it costs somewhere between $800 million to $1.3 billion to bring a new antibiotic to market. Right? And the other thing is, investors have to wait a very long time. So here's an antibiotic that was discovered in 1996, took 18 years to get it into the clinic. Right? So your investors are screaming, where's the money, where's the money? It ain't happening. Right? And if you work on a third world disease like TB, you're not going to make any money from that. Right? Poor people can't afford these drugs. Right? So this is the issue, that we're discovering the things, but to get them FDA approved is very difficult, and it costs a lot of money to develop them. So this just shows our shrinking arsenal, right? So this is drug development. You can see we're just plummeting, right? This should be going like this, but it's going the opposite direction. However, if you want to work on obesity or diabetes, this is where you need to be, because here's a company, right? So these are two drugs. Here's a drug here for diabetes, and there's the other one for an antibiotic. And you can see that the drug for diabetes outcompetes the antibiotic by $11 billion over 5 years. So it's pretty obvious which one you're going to work on, right? You're going to work on this one, right? This is not going to make you any money. And the problem that you've got is you're going to have resistance, right? So there needs to be some incentive. Academics can play a big role here, right? Because big pharma, right, this is hard for them, right? So academics have to become more involved in this research. Right, so I think what we need to do is we need to be developing antibiotics that can be used in animals safely. Right? These antibiotics should not have any analogues in humans. So we can use antibiotics in animals. If the organisms become resistant, it doesn't matter, because those same classes of antibiotics are not being used to treat humans. Right? So what we need to do then is we need to discover new antibiotics for the management of environmental microbes. Right, so I've become very passionate about this. So obviously I was on the other side of the fence throwing stones. Now I've engaged. Right, so this is what we need to do. And we need to partner with industry. Right, so I actually have 3 big programs in my lab in this space. The first is new sanitizers for mastitis. Right, mastitis costs the dairy industry $280 million a year. Right, the best way to combat mastitis is to prevent it. I couldn't believe this. I said, well, how many sanitizers have you got to do this? I thought, oh, well, they'll tell me they've got 10. They've got 2. Chlorhexidine and iodine. That's it. Right? That's what they've got to control mastitis. Right? That's hopeless. Right? So we've partnered with Dearsan, who are the biggest teat spray manufacturers in New Zealand. We've got funding by Agmart, and we're going to develop new sanitizers. Right? Chlorhexidine is on the World Health Organization list as an essential medicine. Right? It's used in 60 different biomedical applications. Right? We need to have new sanitizers in this space, right? Nitrification, nitric oxide emission, greenhouse gas emissions, right? 50% of New Zealand's greenhouse gas emissions come from agriculture, right? The only way to control this is to develop antimicrobials to combat these organisms. So in actual fact, we had an amazing compound that did this, DCD. You spray it on the pastures. As soon as the urine patch hits the soil, this compound here, which is completely harmless, stops the bacteria oxidizing that ammonia to nitric oxide, which is a potent greenhouse gas. Problem was, DCD looks like melamine, and we all know what happened there. Once again, I thought, oh, well, they'll have 10 of these things they can roll out. They don't. They have DCD and one other inhibitor. So now the farmers don't have anything. So that means you can't convert sheep farms to dairy farms. I mean, this is just huge. I just can't, I mean, for me, it's just totally opened my eyes. Right, so we've got investment from Ravens, our Ministry, Prime Minister, to try and develop new inhibitors, and we've actually found new inhibitors that can do this. Right, the other one I've worked on for a period of time is greenhouse gas emissions. Right, ruminant animals, 50 litres of methane an hour, 7 million dairy cows. Do the math. Right, it's potent. Right, we've partnered with AgResearch MPI, and we've actually developed inhibitors. Right, I can't show you, but I can show you the chemist who made them. Right, these inhibitors work in sheep, they work in beef cattle, they reduce methane. And guess what? Animal production has actually increased. Right? So this is where we need to be heading. right? So these are 3 programs that I've got in my lab at the moment. So I'm not going to talk about those. I really want to talk about TB, right? So tuberculosis disease kills 1.5 million people a year. 10 million new cases of TB every year. Of these 10 million new cases, 5% of them are already drug resistant. In China, 30% drug resistance. So 30% of the strains in China are already multi-drug resistant. India, 20%. Right? We have 200,000 Chinese tourists here a year. Right? They're not screened for TB. Right? So this is why we've got to engage globally. Right? We can't stop those strains coming in here. Very difficult to diagnose, incredibly difficult to treat. Right? Difficult to diagnose, difficult to treat, and very, very expensive. This is a warehouse in Myanmar. Right? This was just at one clinic. These are all the drugs in their warehouse to treat multi-drug resistant TB. Right? It's just it's a huge, huge problem. Right? TB is the leading killer of people with HIV. Kills very fast. So you're probably thinking, oh, well, no one's working on TB, no one's doing anything. Well, actually, we're spending $10 billion a year to control TB. Right? The problem that we've got is that the incidence of TB is slowing down. The problem that we've got is that the percentage of those strains that are drug resistant are increasing. That's the issue. Right? So like I said, in China, that's 30%. In New Zealand, I think it's 0.6%. India's 20%. Right? Myanmar's right up there, 30%. So it's not about the incidence, it's just the more of these strains are resistant. That's the problem. Right? And we haven't developed a drug for 40 years for TB. And obviously, Big Pharma don't want to work on drugs for poor people. They can't afford it. Right? So that's where we can come in. So basically, we're fighting a very old disease. People often forget TB killed 1 billion people before we developed the first antimicrobial. Right? So that's a serious disease. So we're fighting TB with all of these old drugs that were developed in the 50s. If you've got TB that's fully susceptible, these are the kinds of drugs that you get. If you get MDR TB, then you start to get injectables. If you get XDR TB, then basically we're starting to run out of options. Now we have totally drug-resistant TB. There are no options. There are no antibiotics. Right? So this is the problem. Sorry about the slide. What we want with TB, of course, is drugs that are not toxic. Many of those old drugs for TB, they would never be FDA approved now. They're too toxic. Right? They make you feel very ill when you're taking them. Right? So toxicity is an issue. They have to be active against MDR and XDR TB. There's no point developing new regimens to treat TB if they can't combat the resistant strains. The other big problem is how do we combat latent TB? 2 billion people have been infected with this organism. Right? So these are the issues that we face. And obviously, the question is how are we going to find these drugs? And for people that have got XDR HIV, this is a race against time for their families. Right? Their life expectancy is very short. We need a game changer. Right? We need to change the game, how we do this. Right? So our hypothesis in our group is that new drugs will be developed through increased understanding of how bacteria metabolize and persist in our bodies, or how they persist in the environment, if we're talking about environmental pathogens, right? So if we can understand that, then we can actually develop new classes of antibiotics. People often forget, in bacteria we have basically 5 essential targets, right? Most bacteria have got about 4,000 genes, and what I'm telling you is that we're only drugging 5 of those essential targets. What we want to do is say, why can't we drug, so for example, most bacteria have a genome of about 4,000 genes, so TB is an example of this, it has 4,000 genes. 614 of those genes are essential for its viability. So then my question is, well, why don't we have 614 targets? Right, so that's what we want to think about. We want to get out of these old targets and get into new targets. But in order to do that, we need to understand what is the organism doing in our body. So TB is a very tricky organism because it has a very diverse pathology. It actually sets up in the very host cells that we use to destroy it. Right? So it's able to disable that machinery and persist in these macrophages. Right? And what you have to do when you have TB drugs, you have to take the antibiotics for 6 months because you have to kill the last remaining cell because some of these cells are non-replicating. Right? So you need a drug that kills a non-replicating cell. Right? So this is a big problem. The good thing here is that this is a metabolic adaptation. These bacteria don't go to sleep, right? They interact with your immune system and they metabolize host lipids. So they're alive, they're viable, which means if we can uncover what those pathways are and develop drugs against them, we can kill them. So that's exactly what we've done. We've figured out that the most crucial part of this organism's physiology, or its Achilles heel, is its energy generating machinery. So the power plant of the cell that keeps the cell alive is its Achilles heel. And so what we did is we basically validated, so this is the machine here, so this is the ATP synthase. This is the machine that the bacterium uses to make its energy. And what you can see here is that this energy generating machine is powered by these other complexes. Right? So this is, I know this is a public lecture, I'll keep it simple, but this machine basically consumes protons from respiration to make energy. And what we've been able to do is to do mutagenesis and validate that many of these complexes are essential for the organism. What we also know is that some of these complexes are important in persistence and resistance to other antibiotics. So we were working on this complex for a number of years, and we were able to show that it was essential for the viability of the organism. Simultaneously, while we were working on this, basically Johnson & Johnson, a company in Belgium, Janssen Pharmaceuticals, were doing a phenotypic screen. So basically what they were doing is they were looking for new molecules that inhibited the growth of tuberculosis. And this is the most significant discovery in TB research in the last 50 years. And what they basically found was that they found this compound here called diroquinolones, and they showed that this compound actually hit the very machine that we were working on. Right? So we'd validated this enzyme as being essential for the viability and the survival of this pathogen, but we didn't have a drug against it. We actually never thought it would be druggable. Right? What Johnson & Johnson did is they found this compound that inhibited the growth of TB, and then what they were able to do is they were able to show through mutagenesis that this drug actually targets the ATP synthase. Right? This revolutionized the way that we treat TB. The reason for this was that this is a target that's never ever been considered for drug discovery in the history of antimicrobial discovery. Right? And the beauty of this phenotypic screening is that the compounds identify the target. Right? We've been fixated on getting a molecular target, getting the high resolution crystal structure, doing in silico docking and trying to find inhibitors like that. That's failed miserably. That's produced nothing. Right? These guys went back and just used compound libraries, just libraries of compounds. They screened this, oh, we've got a compound that inhibits the bug. What does it hit? Hits this target. The reason why this revolutionized the field is that this compound works in 8 weeks. Right? So instead of having a bacterium that we've got to treat for months, 6 months if it's fully susceptible, 24 months if it's drug resistant, this compound is active against all drug resistant strains, right? And it can kill TB, it can achieve sterilization in 8 weeks. So suddenly this drug is the game changer. So now we move from 24 months to 8 weeks. And obviously what we want to do with TB, we want to move to 2 weeks. We want to treat this like any other bacterial infection. Right, so that's our goal. We want to be here, because if we're here, we can probably eradicate TB. New Zealand's got one of the lowest rates of TB in the world, right? But we still need to reduce the incidence by 70-fold to get rid of it. And WHO has designated New Zealand as a country that could become TB-free. Right? That's got to be a goal. To do that, we need better diagnostics and better drugs. We were very lucky, so we collaborated with Con Andres right from the start, because obviously we'd identified the molecular target, validated its essentiality, validated its role in the cell. So we were given the drug from day one. And what that enabled us to do was to partner with Con Andres. Right? What was also amazing is that this drug was fast-tracked. Right? So this drug didn't wait 18 years to get into the clinic. It waited about 8 years. Right? It was actually FDA approved in 2012. And the reason for that is patients with XDR, MDR, TB, they didn't have any options. Right? So the drug does come with a black box label. Right? There's a few things that they don't understand about it. Right? It's a very lipophilic compound. There were unexplained deaths in the trials, but this drug is actually being used in hospitals all over the world, and a lot of clinicians I talk to, they say there's no issues. Bill Denny, very prominent chemist from the University of Auckland, a cancer researcher, he's actually been given the contract from the TB Global Alliance to make analogues of this drug. So this drug is called bedaquiline. Right? And so what Bill's been able to do, he's been able to synthesize analogues of this drug that overcome some of these toxicity problems. Right, so this drug is going to be a massive success, right? It's going to be a blockbuster for Janssen. You talk to Janssen scientists, they tell you, well, the bean counters are telling them every day to stop working on it because they're not going to make any money out of it, right? So it's a humanitarian effort, right? But what's nice to see is that New Zealand has played a major role in bringing this drug into the clinic. So the big question about this drug was, this is the enzyme that it targets, and what it does, it binds to this rotary element of the enzyme. And what it does, it stops the enzyme rotating and therefore stops the enzyme from making ATP. So the molecule goes in here and basically jams the rotation of this little rotary engine. That's how it works, right? And so the working model was that basically the cells run out of ATP and they die, right? But that didn't really explain, right, we've got lots of bacteria that we can deplete them of ATP and they don't die. So what we worked on with Johnson & Johnson was to figure out exactly how this drug works. And so what we're able to do is actually we published with Johnson & Johnson on the bactericidal mode of action of this drug last year, and what we're able to show is that this drug actually kills TB via an uncoupler mechanism. And some of you might recognize that name, uncouplers. So back in the war, when they were handling explosives, a lot of the people used to have chronic weight loss, and the reason for that was their mitochondria was uncoupled. Right? So basically they couldn't produce any energy, so they just wasted away. And in actual fact, some people use this as a diet pill, right? Dinitrophenol. Very dangerous, but if you take low doses of it, you can actually uncouple your mitochondria and you lose weight. So maybe this is going to be a treatment for obesity. I don't know, right? So in any way, bedaquiline has a very similar mode of action, except that it doesn't do it in mitochondria. It only does it specific for TB. So it was very important that we worked out this mode of action to explain how it kills the organism. What we've been able to do is, if we look now at what clinical candidates are coming out, so there's bedaquiline, and if we look at the latest compounds going into clinical trials, they also target other parts of this complex. And what we've been able to show is that these compounds also work as uncouplers, even though they're hitting different molecular targets. Right? So what we need to do then, we know that this is a fantastic pathway for drug development. Bedaquiline tells us this in these new preclinical candidates. So we're actually working on other compounds that hit this, and this is part of my involvement in the Maurice Wilkins Centre. I'll skip over this. So what we rationalised was that this is the structure of bedaquiline. I know this is way too much chemistry, but this part of the molecule here is very important for its mode of action. And what we're able to recognise was that there are other molecules that have a very similar structure. Right, and one of the molecules that we became interested in was this compound called a mylaride. So a mylaride is a diuretic. Right, it actually inhibits an epithelial sodium channel. Right, so it prevents potassium loss but gets rid of sodium. Right, so it's a very potent diuretic. So we thought, well, maybe this molecule here might actually have activity against TB because this group right here is very similar to the most important group in bedaquiline, except this molecule is not very lipophilic compared to bedaquiline. So then we said, okay, let's see if we can do this. And what I can tell you is actually the HRC really liked this grant, and I'll show you why they probably did, and we actually got funded to work on this. Right, so this is a mylaride. It's been used clinically for 30 years to treat, it's a potassium-sparing diuretic, and what it does, it targets this the sodium channel in our kidneys, right? So what that does, it's used to treat hypertension and congestive heart failure. So it blocks this channel, right? So it promotes the loss of sodium and water from the body without depleting potassium, right? So that's very important. You don't want to deplete potassium. So what we put forward to the HRC is, can we selectively optimize our mylaride structure for more potent inhibition of the tuberculosis ATP synthase, right? And this would be a new therapy for TB. Right, the novelty of the chemistry structures would make this class extremely attractive for patenting. Right, it's a new class of molecules. So that's what we did. So there was a mylaride, so we could show that it was very poor inhibitor of TB, about 256 micromolar. This isn't very good, right? But what we're able to show is if we start to make modifications of this molecule, we can get quickly down to 32. We did a whole lot more medchem, and I can't show you the structures, but now we're at 4. Right, so we've gone from 256 to 4 micromolar. So now we're starting to get to the potency that we need to see. And really, we haven't even started this project. What's really cool about these compounds is that they actually inhibit, so there are already strains of TB that are resistant to bedaquiline. These molecules are able to inhibit these strains, right? So they can overcome resistant mechanisms. This is the chemist that I partnered with, a guy called Mike Kelso. He's an associate professor at the University of Wollongong. Right, so, and he was actually working on these compounds as anti-cancer targets. They're very, very effective inhibitors of ATP synthase. They're also bind to the targets. Remember, the target of these molecules of bedaquiline is this rotary element of the ATP synthase, and our molecules are also very effective binders to this target. So we know they inhibit TB, we know they bind to the molecular target with good affinity, we know that they can kill TB inside macrophages, right? So we've now got in vitro activity, and what's really important is they don't have any sodium channel activity, so they're not diuretics anymore, which is important, right? We don't want to be treating someone for TB and the drug having a diuretic effect. So this is very, very exciting. Right, our goal, right, my goal at the University of Otago, my goal in the part of the Morris Wilkins Centre is to get drugs into phase 1. Right, we would like to get a drug made in New Zealand into phase 1 clinical trials. Right, that's the goal of our research. Right, and so the goal of the TB Global Alliance is to get 21 repurposed anti-TB drugs in here by the year 2020. So these are our current drugs in the pipeline, and you can see there's lots and lots of drugs in the discovery phase. So you can see for tuberculosis, even though we've got totally drug-resistant strains, we have a heap of compounds in development, right? A lot of these are going to fail, right? But the more that we can feed in here, then obviously we'll have more success at getting a near therapeutic for this organism. Right, so that's where I'll leave it. So this is my group at the University of Otago, and I've been very fortunate over the last 20 years to have some very good funding. And so I think what this, I remember Warren Tate told me this when I came back to New Zealand. He said to me, if you do good work in New Zealand, you'll get funded and you'll have a fantastic career here. And he was absolutely bang on the money. So I never left, and I've been well funded. And I want to thank you for your time tonight.
So thank you very much, Greg, for a very wide-ranging talk and a very thought-provoking talk. Now, I know that Greg is happy to answer questions, so if anybody has questions, we have a roving mic.
So,
thank you, and that was a great, fascinating talk, and great to see that you've persisted with this research, because there was a while there when you looked as though you were about to give it all up, because you were a bit dispirited about a decade ago. But I'm just wondering, getting back to the animal use of antibiotics, and fantastic that you're doing this exciting new research, but why are we still, using some of these antibiotics not to treat animals when they're sick, but for, just to prevent disease spreading through a flock of chickens, for example? So, especially when you were saying that in some of the research you did a decade ago, there was 97% resistance. So, are there alternatives? Are you looking at things like probiotics instead of antibiotics? Are there, why can't we stop feeding them to literally, for example, to millions of chickens who are not sick? And two other quick questions. Are we looking in the environment, in the waterways, for example, for antibiotic-resistant bacteria, or even, because when you feed them to chickens, they go into the, into the, what's it called? The stuff that the chicken that goes right through into the stuff that is then spread around in the environment, the chicken manure, for example. So are they testing the chicken manure, the water and the waterways, that sort of thing?
I mean, I think, yep, great questions. I think this is really important. So I think there's a couple of things to consider here, right? The first thing is, it doesn't matter where we use antibiotics. We can use them anywhere, and we'll find antibiotic-resistant organisms, right? So that's the first key point. So the second key point is, we don't have a very good understanding about what is the impact of antibiotic-resistant strains in animals, and those animal strains are adapted to animals, and so the big smoking gun is, right, can animal strains, and I could tell you that the number of reports on this, I could count them on one hand, where there's very strong evidence that an antibiotic-resistant organism created in a chicken, a pig, or whatever, went on to cause major disease in a human. And some of that's got to do with the fact that those strains are adapted to animals, and some of those strains are adapted to humans. So the VRE was a fantastic example, right? So I think we had over 300 isolates of vancomycin-resistant enterococci. Right, if we looked at New Zealand hospitals, probably over the same period we've probably had about 20 or 30 isolates. Right? So we had this massive, massive reservoir of vancomycin-resistant organisms, but yet our hospitals didn't have a problem with this. Right? If you went to the US, they never used avapacin. Right? So you would never find vancomycin-resistant enterococci in their broiler production. And if you were to ask me, who's the country in the world that has the biggest problem with VRE? America. Massive VRE in their hospitals. Right? So this was always the issue, that clearly if we feed antibiotics to animals, we'll create antibiotic-resistant organisms. Realistically, those antibiotics are subtherapeutic. What I mean by that is that they are at levels which give the organism a chance to become resistant. Right? When you go to a doctor, he doesn't tell you to go home and take a subtherapeutic dose of the antibiotic. He probably gives you the antibiotic, it's probably 10 or 50 times the MIC. Basically, he wants that antibiotic to absolutely smash that organism so it can't become resistant. So I think that's the issue. And of course, if you go and look in the environment, you're going to find these organisms. You'll find them in the waterways, but what is the true impact of that? And I guess the question is we don't understand what it is, right? I mean, we could, and in Europe, right, there's lots of studies coming out of Europe now where they've stopped using antibiotics, and actually what they show is that the animals do better, right? But they've got to have different farming practices. So I think it's hard to know. And I mean, I think what we were trying to, if you talk to the poultry production, they're just doing what they're allowed to do. They are allowed to do this because math allows them to do this. There's regulations, they have a vet, they prescribe the antibiotics, right? So I think we can't just point the finger and say, oh, there's the problem right there, because in actual fact, what problem has that caused in humans? So yeah, it's a very difficult question. I mean, we've now done deep sequencing on all of those strains. And we look at them and we say, well, man, these strains look the same, but then the strains in humans are not bacitracin resistant. The ones in animals are. So yeah, I mean, probably in the next 6 months we might have a better picture now that we're doing whole genome sequencing on all those animal isolates and all those human isolates over that complete period. So yeah, it's hard to know. I mean, I think, yeah, watch this space. But I think I even saw an article today where I think a company in America now, I mean, places like McDonald's, Kentucky Fried Chicken, they're putting pressure on their suppliers that they will not accept meat from animals that have been fed antibiotics. So I think public can apply this pressure, but if you were to say to me, what's the impact? I don't know. Yeah.
Prof Cook, I really enjoyed this immensely for all sorts of reasons. Having spent 7 years as an HRC board member, it is fantastic to see the impact and the outcome of it. So I'm delighted to have had the chance to come here. I was the world's worst microbiologist ever. I did a degree in botany about a million years ago. My question is, and once again it will show my absolute ignorance, the new candidates that you're developing, what is to stop those then becoming subject to resistance developing despite the very targeted mechanisms that you're looking for?
Yep, so I can tell you how we're going to find out how these drugs, validation of our mechanism of action will be, we'll actually generate resistant isolates in the lab to these compounds. So we can do that in the lab. What we'll do is we'll passage these organisms at subtherapeutic levels, and then we'll look for mutants, and then we'll sequence the genome of those mutants, and hopefully those mutations will map. What they will tell us the types of mutations that could arise when these drugs start to be used in the clinic. So we can generate these mutants in the lab to find this out. I mean, obviously what we've got to make sure when we use these drugs, if these drugs get to be used with TB, one thing that people don't often realise that in TB you're often on 4 drugs, right? It's a cocktail of drugs. And so people have this very simple notion that you take one drug, it hits this target and kills the organism. You have this drug, hits a different target and kills the organism. What happens when you combine these drugs, the mechanism of action is completely different. And that was something that I didn't realise, but the studies are now showing when you put these drugs together, you get a different mechanism of action. You get a lot of off-target effects. So that will be the key for TB. We will not use these amyloid derivatives in a monotherapy. We will use them with other drug combinations that are synergistic, right? So that will be our goal. And obviously we have to reserve these drugs for people that have got drug-resistant TB, because these people don't have any choice. So yeah, we just have to be very careful how we do this. I mean, bedaquiline, there's already resistance. But the problem with bedaquiline, and this is what really irritates me, is that the people doing the trials, they refuse to take bedaquiline and stick it in a new regimen. What they do is they take bedaquiline and they add it to 4 old drugs. And so but they're adding it to regimens where the organisms are already resistant to it. Right? That just makes no sense. And you talk to them and they say, well, you're an idiot. Don't you realize if we change the regimen, this is going to cost billions and billions and billions of dollars. And we can't afford it. So that's what they're doing. They're taking the new drugs and they're adding to them to the old regimens. Right? Okay, they're getting much faster killing, but it makes no sense to me. But that's what they're doing. There's one school of thought that they want to completely change, but this is going to be billions of dollars and it's going to probably take decades. Yeah, so we've just got to be really careful how we use these and not mismanage them.
Professor Cook, just as a follow-up to the last issue, very pleasing to see the sound research that you're actually doing on as far as TB and antibiotic resistance. Now, I just sort of wonder, in terms of other life-threatening diseases, MRSA, other streptococci infection, rheumatic fever, and other problems which New Zealand has, are similar developments in terms of antibiotics happening in New Zealand and elsewhere to overcome, other life-threatening illnesses?
So rheumatic fever, and I was actually just there to talk about this today, the strategies there are not drugs, because that organism remains antibiotic susceptible, can treat it with penicillin. So the key there is, they're basically telling me diagnostics, vaccines, right? I think sanitizers have got to play a big role in there, right? So in terms of group A strep, they don't need drugs, they need these other tools, right? MRSA, drug companies are spending a lot of money in this space. HRC came to the party again, so I don't know if people saw the big story was that one of the last antibiotics that we used to treat multi-drug resistant E. coli was an antibiotic called colistin. Right, it was very toxic. We actually had our first colistin-resistant isolate in pigs, reported in China last year. Right, as soon as they identified that mechanism of resistance, they found out very quickly it was already in humans. Right, so these polymyxin antibiotics that target the cell membrane, which is considered a very poor target. Right, so but we only had colistin left. Well, there's resistance now, and we've gone back and screened some of the freezers in Dunedin Hospital, and actually the colistin-resistant isolates are already there. So HRC just funded a project for Margaret Brimble to develop polymyxin analogues. And what I can tell you is that we've already screened her first 6 analogues, and they actually kill colistin-resistant E. coli. So there's a lot of exciting science here. The chemists are going to be crucial in this development, right? People that know what good drugs look like, what groups of drugs are really important. Biologists need to partner with chemists. Chemists underpin everything, and so that's one good thing. I've never worked with chemists in my whole career, and I've started working with chemists very closely in the last 3 to 4 years, and it's really opened my eyes. So there's a lot of research going in, especially a bug like E. coli, right? Because that's going to make a lot of money for the people working in this area. So yeah, there's research going on.
Thank you very, very much for an enlightening presentation and good luck for success. I guess my concern, and I don't think you're saying this, is that agreed we have to find new drugs, and this is a new drug or a couple of drugs that may be effective against tuberculosis. My concern is that we don't change our behaviour, and you did kind of start to touch on this around prudent use. We use less, we have better infection control, we use vaccines, we have better management in regard to animals, say, that we will just get veterinarians and doctors continuing to use, and the public expecting that, great, this is another drug that's come along, we can continue to abuse it. And you kind of started to answer that in the second to last question.
Yeah, you're absolutely bang on the money. We need to change our behaviour. So I lived in Switzerland for a couple of years. I got sick there a number of times. My family got sick there a number of times. And I can tell you, in the whole 2 years we lived there, not one of us was given an antibiotic. Basically, they just said, no, not giving you an antibiotic. Whereas I can tell you, my family in the last 2 weeks, one of them has been to the doctor and it's come away with an antibiotic. So, and there was no swabbing, no nothing, given an antibiotic because she had green snot. Right, so I think you're right, it's problematic. I'm going down to Gore next Tuesday to talk to the vets down there at Vet South, and then I'm going to go somewhere else down there and talk to more vets. And it's because unless you go out there and talk to these people, I mean, I'm an academic, I sit in my lab and I think they've got these problems and I think I've got these solutions, but I never thought for 1 minute they needed a sanitizer. I thought they needed new antibiotics, and they said, no, we don't, we need sanitizers. So that changed our whole research focus to sanitizers. Right? Forget about antibiotics, we need sanitizers to actually prevent mastitis. Right? So I think it's really good to engage. And I mean, I think what was really disappointing for me 10 years ago, and Sue will back me up on this, we actually had clinicians that weren't supporting us. Clinicians are going, antibiotic resistance, cook, stop pouring petrol on the fire, you're a firebrand, bugger off. And that kind of really, I was really disillusioned by that. And what I've noticed in the last 2 or 3 years, guys like Mark Thomas, Debbie Williamson, these guys have come out on the front foot and said, hey, it's a bloody issue, we need to do something about it. And then suddenly now it's a big problem because now the clinicians are saying it's a big problem. So that irritated me a little bit, but I'm really happy that they suddenly said, yes, it's an issue. But it's not an issue with every single bacterial pathogen. It would be wrong to say, well, every bacterium has become resistant to every single antibiotic. That's not true. Right? TB is a good example because we have strains on the planet that are resistant to all 10 antibiotics. That's an issue. There are no antibiotics for those strains. Right? So that's been really pleasing to see that we've been able to get out there and realise that it's not just about feeding antibiotics to animals.
Yes, I'm interested in the balance between antibiotics and vaccinations. I mean, I understand why it has to be so highly towards vaccinations for viruses because they're using our own metabolism. I'm interested that you've managed to get into the bacterial metabolism that's also central to our own cells and not, not to do with their unique kind of cell wall, but that's another question. But when I was in the third form at school, we all lined up and had our BCG, and I know it's not 100% effective, but has there been decay in the protection of people from TB globally? I mean, it's very depressing that there's just so much of it still when there is a vaccine.
Yeah, but the vaccines, it's the whole lot of populations the vaccine doesn't work on, and so there's been millions and tens and millions of dollars gone into developing new vaccines, and we're still using a vaccine that's probably 100 years old. So that's the issue. And a lot of people have stopped working on vaccines. So there are big groups in the UK still. I mean, vaccines what we want. Like, for example, those environmental issues, right, methanogens, we want a vaccine. Farmers don't want to be putting a bolus of inhibitors into the cow because then Fonterra are going to be complaining about residues. I mean, we want a vaccine, right? That's what we'd love. We'd love vaccines to vaccinate against all these things. But, I mean, for TB, we need vaccines, drugs, and we need diagnostics. Right? Diagnostics alone could save 500,000 lives a year. Right? Being able, because XDR TB, we don't have a diagnostic apart from second-line DST, which is basically phenotypic diagnostic. We want a molecular diagnostic. We've got a molecular diagnostic for MDR TB, but not XDR. So that's the issue. Because if we're able to diagnose that guy in Dunedin Hospital, which took 66 days, if we could diagnose him in 6 hours, then we could have said, right, we need to give you these 5 antibiotics. What we're actually proposing to do, and why we're working in Myanmar, is that we want to use whole genome sequencing. We want you to come into the hospital, we take a sputum sample from you, we amplify the genomic DNA of TB that's in your sputum, we sequence it, and then we'll be able to tell you next morning, you've got a strain that's resistant to these 6 drugs because we know what the mutations are, and we need to give you these 5 antibiotics. That's our dream. We could actually do this in New Zealand because we only get one isolate, we have one isolate of TB a day. We could actually use whole genome sequencing to diagnose drug-resistant TB in New Zealand. We could move towards eradication of TB.
One more question.
Thank you very much for the nice presentation. My question is about India, and it's not one question only because all questions are related. The first thing is you gave the stat about high use of antibiotics in India. Mm-hmm. My question is that used on human or it's also on animals? is the big data?
It's a big number, isn't it?
Yeah.
My understanding, it's human use.
Yeah, human use.
Yep.
Second thing is why the doctors straight away give antibiotics in India because the tests are so expensive. I think it's the cost.
Yep.
Like the TB gold costs 3,000 rupees, which is like $600, you know. And while the antibiotics would cost like 15 rupees or
Right.
100 rupees.
Great point.
So every time you get a cough,
Yep.
they think that, and they straight away give you antibiotics. It could be to treat pneumonia or anything like that, So that is the big problem there, and you can get the antibiotics straight away. Going to the doctor costs money, so you go to the pharmacist straight away. People just buy the antibiotics
Yep.
straight away without even going to the doctor.
Great insight. Yep.
So now the test in Myanmar, is it very expensive, or is it like, when the person comes, and then you tell him this is the genome of the bacteria, and
Yep.
how expensive that test? I mean, how is it going to help the poor countries,
Yeah, so great questions. So, for example, in Myanmar, they have gene expert machines, which are basically molecular-based tools to diagnose drug-resistant TB. This is all funded by the Gates Foundation, WHO. So obviously in Myanmar, no one could afford this, right? So it's all funded internationally, right? And they're rolling out these gene expert machines all throughout Myanmar, right? So that's never going to work. I mean, that will work as long as international people are prepared to fund this, right? But as soon as that dries up, then who knows what's going to happen. So what we're trying to do is we're trying to bring research to Myanmar so that they can, so for example, they couldn't do second-line phenotypic testing there. They can now do this because we've trained them how to do it, right? So this is what we want to do. And it's great that I've got my postdoc who's from Myanmar because he's very passionate about it and he can go back and communicate with the people. But you're right, it's a massive international effort in Myanmar. And because people realise if you don't, it's like the Australians, they're totally engaged globally because the big threat for them is Papua New Guinea. Right, drug-resistant TB in Papua New Guinea is out of control. If you talk to Australian physicians, they'll tell you could go in there with 10 new antibiotics tomorrow, you're not going to have any impact. Right, because you need diagnostics, you need vaccines, you need all three. Yeah, so great points. Yep.
So I think we need to draw the formal part of the talk to a close now, but we would like to invite you to come through and have some refreshments with us, and Professor Cook will be there so you can ask him questions one-on-one. But I'd like you now to just thank Professor Cook again for his stimulating talk.